Thank you, Stefan and Catherine, for the wonderful introduction and for the opportunity to speak today. It truly is an honor to have this opportunity to share with everyone the work we have been doing in Colorado on COVID-19 and metabolomics. And I look forward to hearing from the speakers following me about the applications of their areas of expertise to learning more about this disease. And so today I'm going to tell you about a few of the projects we have been working on related to COVID-19 in studying serum and plasma. And the themes will be around kynurenine pathway, metabolism, and fatty acids. So we are located at the University of Colorado Anschutz Medical Campus, which is in Aurora, Colorado, just east of Denver. We are part of the School of Medicine Metabolomics facility, which is housed within the Department of Biochemistry and Molecular Genetics. And we are a shared resource of the Colorado Cancer Center. And so our objective is to leverage metabolomics, mass spectrometry-based metabolomics, to studying any and all sample types in biomedical applications, everything from blood transfusion to trauma patients, cancer, and now infectious diseases such as COVID. We are led by Angelo D'Alessandro, who is the director of metabolomics and associate professor in our department. He developed a high throughput metabolomics platform that I'm going to talk about in a moment, in conjunction with Travis Nemkov, who is a faculty level researcher in our group. And in addition, we have a team of technicians who do the hands-on day-to-day sample handling work and whose contributions are invaluable in laying the groundwork for the project I'm going to discuss today. Within our facility, we have proteomics and metabolomics applications. We have a variety of instruments. As you can tell here on the left, we have a particular fondness for the thermomass spectrometers. And so the data I'm going to present to you today was acquired on ThermoVanquish UHPLCs coupled to ThermoQ Exactive mass spectrometers. We utilize, as I mentioned, a high throughput metabolomics approach. The rationale for that is twofold. When we think from a scientific perspective, the idea of high throughput allows for minimization of instrument drift, decreases the time from the analysis of the first sample to the very last sample in the queue, which minimizes any changes that might be occurring during the sample waiting time. And then when we think about doing these analyses from a core perspective, the advantage to high throughput is a lower barrier to entry or barrier to investigation. And so this means that submitting labs can submit a greater number of samples, they can have higher N, all of the appropriate controls, and are more likely to generate publication quality data. So what we have found is that even on these very short C18 types of gradients, we can separate metabolites based on their polarity. Shown here is like a 12-minute run, going from the most polar metabolites to the least polar. We published this 2 years ago in Methods in Molecular Biology, and currently we are running a 5-minute method, which is ESI positive. So we run samples in positive and negative modes. With injection time, it's about 12 minutes per sample. And so that means that we can analyze about 100 samples per mass spec per day, including QC and QA runs. And so this really allows for us to run very large sample sets in pretty short periods of time, and then carry forward data analysis with very clinically relevant sample sets. And so today I'm going to talk about applications of high throughput metabolomics in COVID-19. I will discuss our first study, which shows alterations of kynurenine and fatty acid metabolism within the context of IL-6 levels and renal status. And this study was performed in conjunction with our collaborators at Columbia University Medical Center in New York. The second study is one that builds on the first in a much larger cohort. And so there we are able to delve deeper into metabolic heterogeneity of hospitalized patients with and without COVID. And so that study also includes our collaborators at Columbia and then another group of collaborators at the University of Maryland. And the final project I will just touch on is the COVIDome Explorer Researcher Portal. This is a campus-wide effort at the University of Colorado, and it is an online tool for researchers worldwide to access COVID data in not just metabolomics, but other omics types of technologies, where that data can be worked with and downloaded and taken to other platforms. And so to begin, we will look at the effects of COVID-19 on the serum metabolome with a special eye toward IL-6 levels and renal status. And so this study was performed last spring in about April 2020, and these samples were obtained from New York City in March and April 2020, so right when the lockdown was beginning for all of us and when things were particularly bad in New York City. We were fortunate in the sense of that we had a long-going collaboration with researchers at Columbia. And so they are pathologists that had access pretty early on to samples from COVID patients for research purposes. And so we were able to leverage that existing collaboration and Metabolomics platform we already had up and running to begin very quickly analyzing these samples as this was becoming our new world. And so in this study, we enrolled 49 patients. There were 33 COVID positive and 16 COVID negative. The COVID negative patients in this study were either healthy controls or convalescent patients. And so serum was obtained here, and it was analyzed, or I'm sorry, it was extracted via a modified Fulch extraction, which had previously been shown to inactivate coronaviruses. And then the aqueous fraction we analyzed by high throughput metabolomics. I show here the IL-6 levels of the patients enrolled in this study. And so we have control, and then we stratified the COVID positive samples into 3 groups: low, medium, and high IL-6. So IL-6, interleukin-6, is an inflammatory cytokine that is part of the cytokine storm that we associate with COVID. And so as reference, a healthy person, IL-6 levels are around 5 picograms per mil. And so we see, especially with the medium and high IL-6 groups, we see, a drastic increase above normal levels. And in particular, in the high IL-6 group, we can see that there's a subcohort there who, max out the IL-6 assay greater than 300 picograms per mil of IL-6. With the metabolomic profiles, I'm showing here at the top volcano plots, low, medium, and high IL-6 samples versus control. And what we can take home from these plots, which show in red, we see increases in metabolites in the COVID positive. In green, we see decreases in the COVID positive patients. What we can see is that as IL-6 levels are increasing in these patients, we see a greater number of metabolites whose levels are significantly changed versus the healthy control. That effect is also observed in the 3-dimensional PLSDA shown here in the middle. We see the control in the gray dots, and then we see an IL-6 correlated trend away from control. So with increasing IL-6, we see a metabolic phenotype that looks less and less like the control. With untargeted metabolomics data, we did a pathway analysis, and we show on the bottom right that the most enriched and significantly changed pathway was tryptophan metabolism, and also a lot of metabolism pathways of amino acids were enriched and changed when we compare the high IL-6 group to the control group. And so looking closer at tryptophan metabolism, we show here the metabolic pathways downstream of tryptophan, which is an essential amino acid. And so we noticed immediately that the tryptophan levels are decreased in the COVID positive patients, and we see an increase in the downstream product kynurenine in the COVID positive patients. We also see increases in some products downstream of kynurenine, such as kynurenic acid, and we see decreased levels in the COVID patients in serotonin, which is downstream of tryptophan, but in a pathway separate from kynurenine. And so what we can take from this is that in COVID positive patient serum, we see that tryptophan metabolism, the balance between tryptophan to kynurenine versus tryptophan to serotonin has been disrupted, where the pathway that is now favored is tryptophan to kynurenine production. We did a biomarker analysis, so now we've collapsed the IL-6 groups into one, and our question there is COVID negative versus COVID positive. So when we did a biomarker analysis, the question being, are there any small molecule metabolites or which small molecule metabolites are the best predictors of COVID positivity? It turns out that tryptophan and kynurenine are 2 of the top hits. And so we see, using stable isotope labeled standards spiked into every sample, we are able to determine thresholds. So tryptophan was around 100 micromolar in the serum. Kynurenine was around 5 micromolar in the serum that discriminate COVID positive from COVID negative serum with confidence level around 96%. Tryptophan and kynurenine are connected to each other via the activity of indole dioxygenase, or also referred to as tryptophan dioxygenase, TDO. This is the rate-limiting step in tryptophan catabolism. And products such as kynurenine and others downstream are activators of the hydrocarbon receptor, and they are negative regulators of the immune system. And so the idea of tryptophan metabolism being disrupted in a pathological state was something that we had seen before. We have seen this in triple-negative breast cancer. We've seen the TDO axis disrupted. We have seen it in hibernating mammals, such as the Arctic ground squirrel, that tryptophan metabolism changes during torpor and arousal. And we have also seen it in trisomy 21, or Down syndrome. And the link there is that TDO, IDO activity is downstream of interferon signaling. The gene for interferon is on chromosome 21. And so individuals with Down syndrome have 3 copies of the interferon gene, and thus at baseline have higher interferon signaling and higher activity through TDO or IDO. Returning to the story of COVID, when we think about tryptophan metabolism being routed either through serotonin or kynurenine, and we see perhaps a change occurring where more tryptophan is going in the direction of kynurenine, we start to think about big picture. So what does this mean for patients that have COVID? I mean, what does this mean for the immune response? And so one aspect of this, and I show on the right a summary of seroconversion of what happens when we become infected and the immune system begins to respond and grow antibodies. And an important component, without delving too deep in this direction, is platelet activity. And so we see in stage 1 early on a depletion of platelets down on the bottom left. And then in stage 2, in the seropositive state, platelets recover and become activated. And platelet dense granules, as some of us may know, are highly abundant with serotonin. And so serotonin is an important part of platelet activation and platelet dense granules. And so though we have not yet been able to connect all of the dots here, especially with what I have told you so far, I think we can appreciate that when we begin to see these systemic changes and how tryptophan is metabolized and begin to see perhaps a deficiency in serotonin, that we can begin to think about how this can be connected to platelet activity in the context of a process like seroconversion or in the context of coagulation, which we know to be also disrupted in a lot of cases with COVID-19. We also look deeply at fatty acid metabolism in this initial study, and I'm presenting to you just a short summary of what we have seen. And so in the top row of plots, we see individual levels of fatty acids that, in general, the fatty acids are decreased in COVID-19 patients relative to control. And then in contrast, acylcarnitines are increased in the COVID positive patients. So acylcarnitines are the means by which fatty acyl carbons are transported into the mitochondria from the cytosol in order to be oxidized in the TCA cycle. And so when we see systemic levels of fatty acids and acylcarnitines changing in different directions, then we start to think, well, perhaps fatty acids are being used for a different purpose. And so one hypothesis that we can generate from this is that perhaps the fatty acyl carbons are being used in lipid metabolism, perhaps being used to form membranes or for viral replication. We also perform some correlations with clinical markers. So for example, blood urea nitrogen. So we're looking at blood urea nitrogen as a readout for kidney dysfunction. And we see with increased BUN and increased kidney dysfunction, we see increased levels of medium chain carnitines. We performed a correlation with C-reactive protein as a readout for inflammation. And we see with increased inflammation and increased C-reactive protein, we have decreases in levels of a lot of long chain fatty acids, both saturated and unsaturated. And then with IL-6, as I mentioned previously, one of the inflammatory cytokines, we see with increasing IL-6, we have increased levels of kynurenine and some of the carnitines, again, emerging from that analysis. And so in summary from this paper, which we published last year in JCI Insight, we see altered systemic nitrogen metabolism as a key hallmark of COVID-19 infection. We see increased kynurenine and decreased tryptophan as some of the strongest predictors of disease in this cohort. We also see disruptions to fatty acid and acylcarnitine equilibrium and also changes to amino acids. And so from here, we've expanded on this study with additional samples that were obtained later in the spring, so April and May 2020. The idea here is if we can analyze more samples, then we are able to better delve into certain comorbidities, effects of sex and age, and many other factors that are going on in this disease. And so in this study, we analyzed the plasma because one of the major objectives of this study was also to look at coagulation and to correlate metabolite data with coagulation parameters. And so in this study, again, leveraging our collaboration with Columbia, we analyzed 831 plasma samples. From 543 were from COVID positive patients and 288 from COVID negative. Importantly, in this study, these are hospitalized patients who tested negative for COVID. And so that provides an even more robust comparison group rather than healthy controls, because we can be sure that the changes we're seeing are more specific to COVID and not just respiratory illness or severe illness compared to a healthy control. And the other point to mention about timeline is that, these samples were obtained in April, May 2020 from New York City. And so this was before the emergence of the variants that are now seeming to be part of our world and our everyday life. And so this was, before those emerged. And a Folch extraction of plasma and analysis by untargeted and targeted metabolomics of the aqueous fraction. By PLSDA, we show a partially overlapping phenotype of the negative and positive COVID patients. And so this is in many ways expected because these patients are hospitalized and severely ill. Some of them are ICU patients. And so we expect to see a lot of similarities. We looked at kidney dysfunction and dysfunction of many other organs as well, but I'm showing here kidney dysfunction. We see increase looking at blood urea nitrogen and creatinine levels increased in the COVID patients. And then interestingly, since this is a mass spec interested audience, we have creatinine measurements from the CLIA assay performed in the clinical lab. We also have creatinine measurements by mass spectrometry. And so we did a correlation of those measurements and we found 0.87 R-squared value. So we see a nice correlation there illustrating the potential utility of mass spectrometry in the clinic or in the hospital setting to measure not just a small handful of small molecules in the blood, but perhaps hundreds of relevant metabolites in a very short time frame in the blood. On the volcano plot on the far right, we have in the red the increased metabolites in the COVID positive patients, and then in the blue, the metabolites that are increased in the COVID negative patients relative to COVID positive. And so a lot of the same themes are emerging here. We see changes to nitrogen containing metabolites, changes to amino acids, and then an increase in COVID positive patients of creatinine, urate, and kynurenine. Revisiting the tryptophan metabolic pathway, looking at TDO and IDO activity in particular, we see again decrease in tryptophan levels in the COVID positive group and an increase in kynurenine in the COVID positive group, which we measured both by relative quantification levels and then with the spiked in stable isotope labeled standard, we have absolute levels of kynurenine in the patient plasma. We did coagulation assays to look at coagulation phenotypes within this patient population. And though we don't have time to delve deeply into that today, I wanted to present just one aspect of coagulation. So here we did a correlation of the metabolite data to measurements of D-dimer. So D-dimer being a product of fibrinolysis or clot breakdown. And so with high D-dimer levels, we see coagulopathy. So we see that IL-6 is the top correlate with D-dimer. And we see one of the top negative correlates is tryptophan. And so again, we are using this to help begin to understand how disrupted tryptophan metabolism could play a role in coagulation phenotypes, which is just one aspect of what tryptophan and altered tryptophan metabolism could be doing. We then return to the idea of fatty acids. So now with many more patient samples, we are able to focus in on specific time points and specific groups. And so in this presentation of data, we are looking just at the samples acquired at admission of the patients. And so for some of the patients in this study, we have a time course sampling, which we'll talk about more in a moment. But these are just the initial at admission samples. And we've broken the patient groups, we've stratified them by BMI. And so we have from red, the lowest BMI, to pink, the more obese patients. And we see that levels of the free fatty acids here, so these are long chain fatty acids, unsaturated to highly unsaturated fatty acids, we see that they increase with increasing BMI, as we would expect and as is known in the literature. But interestingly, when we compare COVID positive to COVID negative, we see across the fatty acids that those are elevated in the COVID positive patients with respect to the same BMI group in the COVID negative patients. So this is a bit different than the comparison we saw earlier, where the fatty acids decreased relative to control. So it shows the importance of comparing to other patients who are also quite ill. It also shows the importance of time course and trying to match those samples together as close as we can by taking all of the samples from the admission time point. And it shows how there's still a lot more to learn with what is happening with fatty acids, why they are elevated in the COVID positive patients. And as I mentioned, we have a time course on some of the patients within this cohort. And so I just wanted to show one example of how we can use that time course information and longitudinal sampling to compare and contrast. And so on the left, we have a patient who's female, age 14. She is intubated in the ICU and unfortunately had a stroke, but fortunately did survive COVID. On the right side, we have a female patient, age 52, so a good bit older, much higher BMI. She is Black, and so she is in a group that we know is disproportionately affected by this disease. She is also intubated. She suffers from clots and deep vein thrombosis and has a history of lung disease. And unfortunately, she did not survive COVID. When we look at the metabolite data as a function of time, I wanted to focus in on the tryptophan metabolism graphs that I show here. And so we see when we move from left to right, which is our time course, we see, and I should mention that this data is normalized to the first time point, but we see that kynurenine levels very early on, the red trace in the patient on the left, we see that those spike to above twofold change. And though that trace oscillates a bit, we see that those levels are maintained about 2.5-fold approximately over that first time point throughout the patient's stay. At the same time, we see that tryptophan, the dark blue trace, decreases when the kynurenine spikes and stays pretty flat throughout the time course. In contrast, the patient on the right, who unfortunately does not survive, does not have the kynurenine spike that the patient on the left has. Not only does she not have that spike, she does not have kynurenine levels that reach the same amplitude, that 2 and 2.5-fold change over the first time point. And her tryptophan levels continue to increase throughout the time course. And so though this is a 1 versus 1, this shows the ability to generate a hypothesis where perhaps activation of TDO and IDO is important for modulating the immune response in a way that helps us survive the disease, but does not generate such a cytokine storm that it could kill us. We see also important changes in arginine metabolism and fatty acids that I'm not going to go in depth on at the moment, but this paper is deposited in the medRxiv. We deposited it last Friday and it's under review in a journal. So we hope that it should be available on medRxiv in the next 24 to 48 hours with much more information about these time course traces and other data related to these to this cohort. And then the last project I wanted to speak to you about today was the COVIDome Explorer Researcher Portal. And so this was a campus-wide effort at the Anschutz Medical Campus, the mission being to decipher the underpinnings on the molecular level of COVID-19 pathology. So not just to generate a large data set and bring it together and see how it interacts, but then to take that interactive data set and make it available to the general public, or the general scientific community. And so in this effort, there were 105 patients enrolled, 32 COVID negative, 73 COVID positive. They're all hospitalized patients, but importantly, none of them are in the ICU. And so they are, of course, sick enough to be hospitalized, but they are not critically ill that they are in the ICU. And so blood samples were taken. We personally analyzed plasma and red blood cells for metabolomics on these patients. As you can see, there was also proteomics, transcriptomics, and so on. We generated a COVIDome data set, and then that was loaded into a COVIDome Explorer, which is the tool that is freely accessible at the website covidome.org, if you're interested. The COVIDome Explorer workflow, as I mentioned, involves collection of all of these types of data across the top. The data is de-identified and linked together and then viewed through Shiny apps on covidome.org. And so on covidome.org, and I will show a few screenshots in a moment, but there is the ability to see the methods that were used for this analysis, publications that will result, and then ability to perform different types of analyses. So statistical analyses, visualizations, ability to interface with other databases, and then at the end, download the visualizations or the raw data files in order to import in any software of your choosing for data analysis. And so here's a screenshot of what the COVIDome Explorer looks like. I'm selected right now on the Metabolome tab, since this is a metabolomics interested audience. And so it has a brief description for people who are new to this area of research. So what is the metabolome and why is it important? Which methods were used to collect this data? And then once you move into the Explorer, you can see I've chosen here on the left that I'm interested in the effect of COVID-19 status. You can also look at the effect of seroconversion. And so on the left, there are many different menu options. There are statistical analyses that can be performed. And so for a simple example, I'm showing plasma looking at males 21 and over. And from the drop-down box, I've chosen the metabolite fumarate. And so we see in the volcano plot in the center here that fumarate is a metabolite that is increased in COVID-19 positivity. And on the right, we see a box and whisker plot with superimposed dot plot of the fumarate levels in the COVID patients. And so we can see how that's elevated in the COVID positives. We can see the spread, and we can see a p-value of 0.02. And so this data these plots can be exported, the raw peak areas can be exported and then imported into software of your choosing. And so this is a paper we have under review in a peer-reviewed journal, but it is also deposited in the medRxiv. And I thought maybe the easiest way for any who are interested to access it is through the PMID shown here. And so in summary, we show that systemic metabolic profiles characterize the small molecule phenotype of COVID-19. We see a lot of changes to nitrogen metabolites in circulation. We see activation of the kynurenine axis, and we see what I am generalizing as mobilization of fatty acids. So depending on the exact context of fatty acids, they can be decreasing or increasing depending what we're comparing them to. But we see changes in fatty acids that can tell us a lot about lipid metabolism. It tells us about how the virus is using the host cells to replicate and how process is causing changes in systemic metabolism. And then in the larger context, I have shown the utility of high throughput metabolomics in large clinical sample sets. And so this means that we can take projects of interest from clinical trials to infectious disease, blood transfusion, aging, exercise, and we can tailor extraction and acquisition methods to the scientific questions of interest. And then we can annotate with metabolites and validate through standard protocols. But then we can take these features and these named metabolites that I show here at the bottom in the gray dots, and then we can begin to stratify the cohorts. We can break them down by sex and age. We can look at environmental factors and genetics factors, and we can begin to connect the dots and build the puzzle and try to understand where to look next in terms of a mechanistic sense for building a big picture of how metabolomics is changing within samples and what that means for a disease state or a process such as aging. And so with that, I would like to thank everyone who is involved in this work, my lab, everyone at CU Anschutz, including Joaquin Espinosa's group, who was the primary driver of the COVIDome Explorer portal, our friends at Columbia, University of Maryland, and University of Virginia, in particular, Steve Spitalnik, Tiffany Thomas, Richard Francis, Eldad Hode, Paul Bueller, Jim Zimring, and then our sources of funding. And as I'm sure you all can share in with me, we did not have funding to do COVID work when we began doing COVID work. And so we are grateful for our financial support from the National Cancer Institute, NHLBI for our work on blood transfusion, and NIGMS from our work on trauma, which have allowed us to have these existing collaborations and have metabolomics platforms up and running so that we can quickly pivot to a brand new infectious disease and do our best to help the community learn new things. And so with that, I thank you all for your kind attention, and I would be happy to take any questions or feedback that you have to offer. Thank you very much.